Okay, so welcome everyone to this innovation workshop, part of the Genesis conference, the innovation to deliver sustainable, consistent reagents from research through the clinic, proposed by Abcam. So in the next hour, you will hear from 3 Abcam experts on that topic, and you will be able to ask questions at the end of it. So as a starting point, if you can keep microphone and camera off, and once ready, just to put question in the chat box and we'll take them toward the end. So our first speaker is Dr. Will Howard. Will leads the Abcam Global Imaging and Immunohistochemistry team as Director of Antibody Validation. Prior to joining Abcam in 2018, he worked at AstraZeneca as a team leader of its molecular pathology group, where his team developed biomarker assays for preclinical phase 1 and phase 2 clinical trials. Before that, he was at the CRU Gates Cambridge Institute, where he set up and ran the histopathology ISH core facility for 9 years. He has also worked at the Wellcome Sanger Institute on the Atlas of Protein Expression and was responsible for R&D within the immunohistochemistry group. So I'm going to hand over to Will to start his presentation. Will, go ahead.
Thank you very much, Eileen. I'll just start the presentation. So welcome everybody, and thank you for joining this one Nucleus Innovation Workshop. As Eileen's already highlighted, I'm Will. I'm one of 3 presenters today, and the others you can see Dr. Paul Wiley and Dr. Gráinne Dunleavy, who will tag team our way through this talk on the innovation to deliver sustainable and consistent reagents from research through to the clinic. So obviously we're Abcam. It won't surprise you to know that antibodies are at the center of our reagent development. And they're at the center because obviously we've got a whole host of pieces available to us. We use antibodies, obviously, as part of our antibody discovery, but I want to highlight to you the additional things that are present, which is that we have some very high quality recombinant proteins which are used in part of the antibody discovery process, but are also available, which Gráinne will talk through in a few moments. We have a whole host of cellular editing processes through CRISPR, which we'll have a bit more discussion, used for validation, but also therefore available for performing cellular assays, epigenetics, for example. But you know, we've also, once we've got an antibody, we've got the capabilities to add conjugation and labeling to that, and once you've got that conjugation, obviously you can lend yourself into that singleplex and multiplex immunoassay arena, and all of these form a feature of this talk today. And we've been doing this for a while, so you know, we 20 plus years of running antibodies through pipelines at scale, and you know, the general way of looking at it is we start with a target, we start with protein design, and that protein expression and QC, very high quality proteins, leads to very high quality antibodies. So you lead into antibody development and production, and of course leads into this very vital part, which is the characterization of the antibody to really demonstrate that the antibody is binding to the thing that you originally set out to do. And finally, at the end is that batch-to-batch QC and the ability to make sure that we are producing reproducible antibodies, providing that out to yourselves. And so what we'll do is as we work our way through the talk, I will now hand this over to Dr. Gráinne Dunleavy, who will talk you through the proteins aspects of this process.
Thanks, Will.
Thank you, Will. So our next speaker is Dr. Gráinne Dunleavy, who is the head of protein science at Abcam. In her current role, she focuses on protein production for in-house use and catalog sales. Through his work, she has led process improvement that allowed Abcam to consistently produce high-quality proteins in a high-throughput production process. Gráinne, over to you.
Sorry, it's just, there we go. So as Will said, all good antibodies start with a high quality antigen. And when I started at Abcam, I was tasked with improving the efficiency of our antigen production process, whilst also increasing the quality of those antigens and giving us the ability to make more really tricky proteins. So that is obviously quite a challenge, but during this talk, I'm going to introduce you to the recombinant protein production platform that resulted from that piece of work and the new range of premium bioactive proteins that we have launched and why we've decided to launch them. So high throughput protein production obviously provides many challenges. And if you think about the antibody, which is Abcam's workhorse protein shown here on the left, the majority of that protein is the same. Obviously it has some changes in the CDRs which come with their own individual production challenges, but for the most part, the protein production is relatively similar from one antibody to the next. The rest of the protein universe is obviously quite diverse, and so each individual protein comes with its own unique challenges. So trying to make a process where we produce lots of these proteins and we produce over 1,000 high quality recombinant proteins for immunogens every year means that we have some challenges to overcome in trying to produce them in a more streamlined process. So how do we do this? Well, we start by breaking down the process into individual parts. And the first part is protein design. And the next stage, and we use obviously bioinformatics to give us the best sequence for both expression and for immunization. And the next part is expression screening. So we introduced this expression screening step in high throughput because every good protein production starts with a really good expression. And so the better the expression, the easier it is to produce the protein downstream. The next part is protein production. And in our protein production, I'll show you how we do a high throughput process, but we also have run in parallel a protein production process where we can use individualized methods for production of those proteins when required. Moving on from there, we have our quality control, and we have quite a stringent quality control in place. And finally, fill and finish. I'm sure you're familiar with that, so I'm not going to give much details on how we finish the protein, but I'm going to now talk to you in detail about each of the other stages of that process, so protein design, expression screening, production, and quality control. So starting with the protein design, as I say, we use bioinformatics to tell which is our best immunogen, and also to look at our solubility and think about how expressible it will be. But it is often difficult to think about which construct we should go with. We can predict the best protein for expression, but is it always the best immunogen, or will the best immunogen express? The next part is thinking about the vector that we should use for that protein expression. Thinking about the promoter, what signal peptide should be in there, and so on. And finally, the choice of tag. We can add a solubility tag to boost expression levels and solubility, but do we always need it? And if in fact we add it, it adds an extra process step, which increases the time for production and also reduces our yield overall. So we only want to add that where it's necessary. These questions can all be answered through our expression screening platform. In our expression screening platform, we screen all the different constructs and the different vectors of interest in parallel in just 0.7 mil expression scale. We have our DNA synthesized externally, and then we quantify that in high throughput 96-well format. We use the information from the DNA quantification to set up our transfections or transformations using liquid handling platform. And then, as I say, we express that in 0.7 mil scale. We express it in a couple of different conditions at a time, and then go forward and purify and analyze that protein. I'm often asked about the scalability of our process. Obviously, we're expressing in 0.7 mil scale in either HEC or E. coli for our standard process. And people often wonder, can we scale that up to produce our protein for immunization? So we scale up to 1.6 liters. And as you can see, we've worked on this process to optimize it, and we consistently now get over 90% consistency between our 0.7 mil scale and our 1.6 liter scale. As I say, we use just a couple of conditions in our standard process, but that doesn't suit every protein. And so we also use that same expression screening process to run a variety of other conditions. So looking at feed and media, time, temperature, even CO2 concentration. And we use statistical analysis of those results to predict what the best expression condition would be. Moving on from there, obviously we need to scale up, and as I say, we have a high throughput production system. In a high throughput production, we need to keep many things the same. So it uses, in this protein maker, the same size column, the same volume of load, 1.6 liters, the same buffers. And we run them all in parallel, and that suits the majority of proteins that we produce. However, there are more challenging proteins, and we obviously have the ability at the expression screening stage to decide what the best expression conditions for those proteins are. And so they need to be treated individually. And we have a team of scientists who can do that, and who then take those individual proteins, think about their PI, what buffers they need, what the best time of expression is, if they need a different column, and so on. And we have equipment such as this ActiveAnt, where we can screen a variety of different columns and a variety of different buffers to give us the best purification method for that protein, keeping it stable and intact through our process. And finally, we have our protein analytics. Now we use our protein analytics really for two things in this process. One is for the quality control at the end, but the other thing we use it for is thinking about any problems that we may have with those more challenging proteins. So for example, if we're seeing cleavage from intracellular proteases, we will see that through mass spec. Or if we are storing it in the incorrect buffer, we will see that through our DLS and SLS profiles. And obviously measuring the purity with HPLC as well. Now, as you can see, we've developed quite a good skill set for protein production, producing those high quality antigens that we need for our recombinant, for our monoclonal antibody selection process. And we then realized that we have a skill set that is important for production of proteins to catalogue. So summarizing on our Immunogen production summary, we use bioinformatics to inform the construct design. As I say, all good protein production start with a good expression, so we introduce the expression screening. Then we have our large scale purification, where we can either do in high throughput or with individual attention, so we're quite versatile. And then we have our stringent quality control. When we think about products on the catalogue, we come across lots of publications such as this one, where they talk about reproducible data and data that was not reproducible and how much money is spent on that. And of the experiments that are not reproducible, 36% was spent on reagents that were not fit for purpose. And some of these reagents are in fact recombinant proteins. And if we think about why, sometimes coming to a catalogue, it's quite difficult to select which protein you want, because there is the QC on the data sheet is lacking, and you can't see that the protein can deliver what you need to. And in fact, as an in-house protein scientist in biotech and biopharm, I was often asked for a much higher quality of protein to be produced in-house than the proteins that scientists were happy to buy from the catalogue, which for me at the time, a little bit miffed, because it was much more difficult for me to produce the proteins I was being asked for. But obviously people were asking for that because that's what they really needed. And also if they were buying in, would buy in a small batch of multiple different proteins and QC them in-house, because the QC was lacking on the catalogue. So obviously there is a need to deliver a range of proteins with the QC that we deliver for in-house proteins. And so we've launched a new range of premium bioactive proteins that meets those QC standards. And the first range are cytokines, and they're aimed at cell culture. And to add an extra challenge to this, we decided that all the premium bioactive proteins should be produced in mammalian expression system and animal-free. And I'm sure you're aware that cytokines can be quite difficult to produce in mammalian expression system. But this gives us the advantage for a secreted protein that these proteins will get the correct post-translational modifications. And so they are more stable and can be more active, depending on the protein and what PTM it required. They're also tag-free. Sometimes tags can interfere with activity or stability. So these proteins are all tag-free, so they're basically in their native form. They have ultra-low endotoxin because, as you know, endotoxin can have quite a profound effect on certain cell types. The activity has been confirmed. The purity is confirmed by HPLC, which is quite a stringent QC measure. And the mass is analysed by mass spec, meaning that the identity is confirmed and we know that there is no potential cleavage there. And finally, we deliver batch-to-batch consistency, so your results will be reproducible because we use such stringent QC parameters that once we produce a new batch of protein, we know it will behave in exactly the same way as the last batch. So I'll give you some data from these proteins that we have produced. So this is just a typical example. This is some QC data from human TNF-alpha. On the left-hand side, you can see the activity assay, and it's performing as expected in a dose-dependent apoptosis assay. In the center there, you can see the HPLC and the STS page confirming the purity and that it is of the expected size, etc. The endotoxin level being 0.0049, our cutoff is 0.005 EUs per microgram. You will know, I'm sure, that many of the proteins available have 1 EU per microgram as their standard on catalogues. So this is much lower, and obviously that's so that it will have, there won't be an effect of the endotoxin on the cells. And finally, on the right-hand side there, you can see the mass spec confirming that the protein is, in fact, of the mass we would expect. So that shows again the identity. So I've promised you batch-to-batch consistency, and so obviously we have actually tested this out in-house. So we produced several batches of protein on several occasions, and then wanted to make sure that this data lines up. And you can see there the retention time in the reverse phase being pretty much identical, and certainly within the error of the instruments for each batch measured. The mass spec data, again, being within the error of the instrument. And the assay data, the assays were run side by side, and you can see that the curves pretty much overlay in each case, showing that they are equally active to each other and behaving in the same way. So we're pretty happy with that. But we wanted to see as well if the proteins, how the proteins measured up to proteins that you could buy from other providers. And obviously we didn't want to generate this data ourselves. We wanted someone else to validate this for us. And so we went to Genoway and we asked them to buy in some proteins from competitors and run our proteins alongside to see how it would measure up. And you can see in each case that Abcam's protein is either equivalent or slightly more active than the competitor ones that were measured. So we're pretty happy with that data as well. So you can, I hope I've showed you that it's important to QC the proteins as we would have for in-house proteins, and we're noticing an increased demand for this higher level of protein QC than was there previously. And so this new range of premium bioactive proteins has been launched to meet that demand. And in conclusion, we've set up a process to deliver a diverse range of challenging proteins as immunogens. And a key part of that is getting the right expressions or expression screening system. And we've used this knowledge then to create the premium bioactive proteins and other proteins of interest. So I'll hand over back to Will now.
Thank you, Grania. So I will take you through now areas around antibodies. So as we move through this process from the proteins through to the generation of the antibodies, as you know, there are a number of ways of generating antibodies, whether through the standardized or way of immunizing an animal, be that a rabbit in our case or a mouse generating immune response, and then whether you can go through an isolation of the circulating B cells, or create your own hybridoma, which is fairly standard methodology. There's of course the alternative naive approach, creating an immune library, in this case a phase display library. Ultimately, for Abcam, where we want to be sitting is here, which is the generation of recombinant antibodies. So, and by doing that, what we mean is that we take the individual sequences of those clones and we take the CDR, we understand exactly where that is, and that means that we can then in vitro produce the antibody in our own hands, and we're not then reliant on a hybridoma production. Why is that important? Well, the advantage of those recombinant monoclonal antibodies, obviously monoclonal, we've got a single epitope, that stands to reason. But because of that consistency in that we are transfecting in a plasmid into HEC293 cells, we can do that as a stable, as a transient transfection. We can therefore get a better improved consistency and reliability. And our batch-to-batch variability is lower. We're not relying on the continual growth of a hybridoma. And you can see on the left-hand side there, 6 different batches of antibody. In this case is a PD-L1 antibody 288. In immunohistochemistry on positive control of placenta tissue, and you can see that the variation is minimal between those individual batches. But what you don't see is the improvement in that sensitivity and the specificity. And what happens in a cellular system with a hybridoma is over time, mutations are ongoing, and you can often get a slow creep of that hybridoma. Either it changes slightly in characterization or it induces more background. Whereas when you're in a recombinant phase, then that doesn't happen. You know exactly what you're putting in every case, and you're producing that antibody exactly as specified. And so, and the other part of that is it's very highly scalable, and in vitro production, it's grown in flasks, so it makes it a lot easier. And we want to be in that space, and we want to be out of the polyclonal space for exactly the reasons that are on this slide. So on the left-hand side, you can see 8 batches of the same polyclonal antibody, and it's not that they're not specific, which they are. But individually they are different. And so every time you produce a polyclonal antibody, you have to revalidate it or retest it to make sure that you're getting the right quality. Whereas on the right-hand panel, you can see a recombinant monoclonal for the same target with a knockout in all cases. And you can see that it takes that problem away of having to keep on reproducibly testing all of your batches in every cases, and that batch-to-batch variability disappears. So once we've got an antibody and we know that it's high quality in its production, we need to be able to do the validation, and that validation is very important. That validation has to be tailored. So we will tailor that validation as required. We'll, in the production of the antibody all the way through the process. For example, histone antibody, we will run a peptide array. For in this example, the Ki-67, we wanted to run a flow cytometry and an ICC. It'll also be validated in ICC. In each case where it's relevant for that target, we will run as many applications as we can. And we'll do that both in a human space as well as on a mouse rat, so that we can be confident that when we put this onto the website that everybody looks at, and it states it works in a particular application, then it genuinely does. And that can be seen in these particular examples. In an immunohistochemistry area, we will run a tissue microarray. There'll be a multi-tissue tissue microarray, multi-human normal, a multi-human tumor. So multiple different tumor types. There'll be 2 cores of each. So we know we're getting high quality in there. We know we've got certain positives. In this case, the neural tissue is coming up for MBP, and all the other tissues are going to be negative. And we'll also run a mouse rat TMA again, so we've got confidence that when we state that it works on another species, that it genuinely works. But then we can also do some extension work. So not only will we do the standard work, but we can extend where it's relevant to that particular target, extend out into a disease, an indication of interest. In this case, again, PD-L1 put onto a non-small cell lung cancer TNA is about 160 cores to demonstrate the sensitivity of the assay. And this is a fully automated IC assay. And we can extend that further. For example, this particular clone will then run on a cell pellet. So cell pellets are generated by histocyte laboratories, the bottom corner, of a 3+2+1+ and a negative cell line. So you can see again the quality of the antibody through that validation. Of course, the bit that's coming to, which you would expect to have here, is where's the knockout? And so we've been working in knockout since 2015, and we're very proud of it. And there are a number of different ways that we've been doing this, mainly through the CRISPR area here, but there's a number of different things that we can align in here, not just around knockout, but other ways of validating, whether that's through transgenic models, knockout animals, whether it's RNAi instead of CRISPR, that tends to be less popular these days. But we also do cDNA overexpression where required, and we're starting to extend ourselves into looking at protein interference inside the cell by use of trim away or some other technology. But knockout is the main lifeblood of what we do. We use it routinely in the production of our antibodies, and I'll come and show you some examples of that. I don't really need to go through this slide. I'm certain that all of you understand how CRISPR-Cas9 works. And we started off on the right-hand side using a haploid cell model, and we're moving more, as we now have in-house editing capabilities, of using diploid cell lines more akin to the cell lines that are being used in the lab. And so, this allows us to be able to give us confidence that the antibody is binding to what we think it is, whether that's in an immunocytochemistry assay or in a flow cytometry assay. Primarily, we will be using a Western blot. And this is a great example of using those knockout cell lines, and lysates in this case, to validate the antibodies that are on the catalogue, so you have confidence that we know that they work. And so you've got 5 antibodies here, and you can focus on the one on the right. It's a rabbit polyclonal, and the Western blot, if you are getting it a standard Western blot, it looks like it's at the right band size. It's at 39 kilodaltons, give or take. You got that result, you'd be happy. Apart from when you run the knockout alongside, you can see clearly there's a band in the knockout, and it's non-specific. And that's why we're doing this, and that's why we're doing this for over 3,000 antibodies to date. And it gives us confidence that we know the products that are present in the catalogue, particularly where they're labeled with knockout validation. It gives you confidence that that we've done all the validation needed in order to be able to make sure it works. Of course, that product's been removed from the catalogue and you can no longer buy it. But it's not just there. I mean, those gene edited knockout cells are used to create lysates. You can buy those lysates. We use those lysates internally for validation too. But actually, you can use those lysates in your own hands, or you can get the cell lines yourself in order to be able to do your research. So you can use them for your own biomarker discovery, for your hypothesis testing within your own hands. And we can't make knockouts for everything, so there is the option there that if you so wish, then we can do custom gene editing for you, because we now have those in-house technologies available to us. So we can extend that out for you to be able to get you to where you need to be. So having finished up on the fact that what we go to in order to be able to create these antibodies from a validation perspective and to be confident that they work on the things that we think they work, we then want to move on to the things you can do with them, and that's where we're now moving into multiplexing arenas. And I'd like to highlight a couple of slides to you around where we extend. Now we know the antibodies work, we can extend out into a multiplexing environment. In this case, it's a 6-plex. Again, it's immunohistochemistry, 6 different targets plus DAPI, and you can see the quality of the imagery that you're able to generate, and it can reveal some hidden value. But I think you can all agree that there's no real hidden value in a tonsil. But there's hidden value in this. So you extend this out into a lymphoma TMA. There's about 100 cores in this TMA, and you start to see different patterns. So different patients react differently, and in this case, I've shown you 4 individual patterns that have come up on this TMA, again stained with the same 6-plex panel on the left-hand side here. But what you're seeing is some patients are more like a CD3 high, PD-L1 high phenotype, and some patients are more reverse, having very macrophage high, CD68 positive, PD-L1 high. But equally, there are some relatively low abundant things going on. These are largely CD68 positive, CD67 positive, and the bottom right is more CD68 positive, CD3 positive. But all of these are within this lymphoma patient cohort, and that's the power of the multiplex, is allowing you to drill into that, albeit in this case in a 6-plex panel. But you can extend that out. So, so as you're probably already aware that we have an arrangement, an alliance with NanoString, whereby you can come to Abcam and leverage the power of those 4,000 IHC validated RABMAB antibodies and apply them within a geomics assay. 5 of those geomic spaces are available for those antibodies, and the other 90 are as part of that panel from NanoString, so you can create a 95 data set. And those will be custom conjugated for you, they'll be QC tested, and then they'll be supplied to you from Abcam. So you've got a whole host of things that you might want to add into your data set, because that's where your research is taking you. And that's the power of that alliance between ourselves and NanoString. And that's where I will end this particular part, and I'll hand it on to my colleague, Dr. Paul Wiley, who will go into more multiplexing, again with the quality of the antibody as a backstop to knowing that the assay will work for you.
Thanks, Will. So I want to just take my section to talk about multiplexing within a biomarker discovery. So building on what Will's just told you about the work we do for the recombinant antibody technology, we've now utilised that to use the pairs in the same format and the same quality to produce highly multiplex biomarker assays. So we use at the moment a system called FirePlex. This is a technology that came out of MIT around 10 years ago and uses optical liquid stamping of hydrogel particles to create this platform that we use for biomarker discovery. We have it in 2 key formats. One is one in a flow cytometer, is high-plex, so you can do up to 70 different microRNAs or protein analytes, and I'm going to concentrate on proteins today. So that's 70 analytes per sample per well, with very minimal sample volumes required. And then we extended that out to a higher throughput version, which is run on 384 well plates. So this is now a no-wash automated assay, where you can run 384 samples in a very simple automated fashion. And we're about to release in January 1536 well format of that for real true high throughput screening for the biopharma market. So the FirePlex platform itself, the core of it is a fluorescently encoded hydrogel particle. And you can see in the diagram on the left, there's 3 key areas. There's the ends, which are in green and yellow, and that represents different intensities of green and yellow dyes. And because there's different codings at different ends, it allows us to generate up to 70 different distinct codes. And then in the central area, we attach our capture antibody, and this is where the analyte quantitation region is, using standard sandwich immunoassay principles. In the middle here, we have regions where there isn't any dye at all. So this gives us a spatial difference between where the coding happens and where the analyte detection region is. And that's unique compared to most other platforms where the coding and the analyte detection is done at the same time. So this is a really key part which prevents bleed and interference during our data acquisition process. As I said, the FirePlex particles are made of PEG, which is biologically inert, and that enables the analyte quantitation directly from complex matrices. I'm going to show some data on that later on. But we use things like serum, plasma, and CSF, along with more common platforms such as cell supernatants and lysates. And this is without any matrix effect or autofluorescence of the particles due to this unique structure. Also, because of the way we use the PEG, it means our capture area is larger because we can actually bind more capture antibodies to the particle itself. So this increases our sensitivity of data that we can get. So that's the fundamental of where the platform sits, but it's all about actually the antibody pairs. This is where the real content is key. So as Will described, we have a very precise and quality-driven process for generating our antibodies. And it's no different when we're doing pair development. So we use those same antibodies we have pairs here, and then they're used in all of our paired assays, whether it's just a matched pair that people can buy to build their own assays up, whether we use it for our simple step ELISAs, which is our simple 90-minute protocol for ELISA, or for all our multiplex immunoassays. And we go through a huge amount of validation development to make sure that our pairs work together. One of the reasons for this is when people start to look at doing multiplex assays for the first time, the biggest fear that they have is that there's going to be a lot of development for them to implement that into their laboratory. So by doing this work in the first place, we take that work away, and then the kits that we produce can be used with very little validation. So the key areas we look at are things like dynamic range and sensitivity, and I'll talk a little bit about that in a second, but also things like precision. We have protein standard curves in there, which we compare against world's WHO and NISBC standards. We do assays such as spike recovery, spike linearity. So we go through this whole process to really make sure that we have a really good, well-validated assay. And because of the recombinant technology, as Will described earlier, it allows us to know that between batch to batch, the data is going to be very, very similar. So there's no real requirement to revalidate between different batches. One of the key parts of doing any multiplex assay is pair optimization. You can have really great antibody pairs, but sometimes they obviously have cross-reactivity issues. So here you can see the kind of work we do for pair optimization. So we always calibrate our Immunogen or to commercial Immunogen or commercial protein or biologically active protein, just to benchmark against the literature as well. So we know that what we're getting is what we should be seeing. We optimize our assay to make sure the lowest standard for signal to noise is 1.3. And as you can see on the graph on the right, which is where we finished, we avoid the plateau at the top of the curve that you might see here. So we develop it to make sure that the range of the assay is in the linear part of the curve. And we also optimize our pairs for the lowest MDD as well. And so the other thing we also do for our flow-based assay, which is a washed format, that's the 70-plex washed assay, we make sure we have an 8-point standard curve. And then for our no-wash, high-throughput 3 to 4 based assay, we optimize it for a 5 to 6-point standard curve. So these really give an example of where we start with our initial testing against certain curves, and then by the time we finish, we have these really nice, robust data sets that we know are going to be reliable and robust and give the customer good quality. So here's some example data from one of our panels. This is a 17-plex cytokine panel. On our website, we always publish our data, so you can always see our dynamic range and sensitivity. And you can see in this example, we have a sensitivity range from around about 0.1 to 3 picogram per mil, with a dynamic range of up to 5 logs. So it's a very strong, robust assay. And you can see here, these are the individual data for each analyte on the left, and our data against a standard competitor from our benchmarking studies. And you can see in a lot of these samples, we're a lot more sensitive than systems. We also have a 70-plex discovery panel. We have one for both human and mouse. And this is really good for identifying in an early stage project which biomarkers are being upregulated or downregulated that may be of interest in future projects. So you can see this whole range of analytes here. And again, you can see one of the key parts of this is all the curves are a straight line. There's no saturation at the top. So always within that whole range, we are within the dynamic range of the assay. That's a really important component of this. We also then have the ability that once somebody's run this discovery panel, because we know that there's no cross-reactivity issues with these pairs, it means that customers can then go and pick individual ones of these, and we can build them for somebody, and we know they're pre-validated because we know there's no cross-reactivity issues. So then typically somebody will then go and do a larger study picking out analytes of interest within these panels. So I said earlier, we validate this in a range of biofluids, and that's part of the benefit of the particle technology we have. So in this example here, we're looking at the expression of all these cytokines, but evaluated in a range of native biofluids, such as plasma, serum, CSF, and urine. And you can see that there's really good sensitivity within these different tissue types. Again, in this one, we're now looking at different tissue formats. So this is using mouse and human discovery panel. And again, you can see here using either kidney, liver, or lung in the mouse model, or lung lysate, liver lysate, or heart lysate in the human model. We're getting nice data across that sample. So we're getting really good data within these very complex matrices that often are problematic for people to run in a multiplex high throughput format. And then finally, just a little bit of data on our 384 well assay. So this is a no-wash assay that uses the high content image as a readout. And it literally is add the components, add the sample, leave it overnight and read it. And you can see here, we're getting a nice curve on a 10-plex human cytokine panel. And again, here's our sensitivities for our panels. And in terms of cross-reactivity, to see, we get excellent data here as well. So from a screening perspective, for if people want to really start running high throughput versions of this, then this is a really good platform to do that, especially with that consistency over batch to batch. So running samples over long periods of time is really advantageous with the quality of antibodies. And I just want to finish with a case study of somebody who's run the HT format, so the 3-4 well-known. This was published by Goubier et al. From Roche in Basel just this year. So the intro to what they did is they were looking at macrophages and producing macrophages. So tissue-resident macrophages are key players in inflammatory processes, and their activation and functionality is critical to health and disease. So they're a key target, therefore, in drug discovery processes due to that major regulatory role they have in health and disease. And currently, the main source of macrophages used for therapeutic compound screening are primary cells that are isolated from blood or tissue, or immortalized on neoplastic cell lines such as THP1. So it can be quite limiting in how many samples or compound samples you can run using that process. So what the researchers did in this study was to generate induced pluripotent stem cells for the high yield, large scale production of cells resembling tissue resident macrophages. So the iPSC derived macrophage-like cells were thoroughly characterized to confirm their cell identity and their suitability for drug screening purposes. And these iPSC derived macrophages showed a strong cellular identity with primary macrophages and recapitulated key functional characteristics, including cytokine release, phagocytosis, and chemotaxis. So among the other tools the study did, they used the FirePlex-HT assay to look at the released cytokines from the cells they generated compared to standard cells they were using in the past. So briefly, this is the process they were looking at. This is a scheme of the prolonged activation of macrophage progenitors in suspension culture. The suspension culture here allows the accumulation of several harvests over periods of weeks, and then the start of macrophage differentiation from a large homogeneous population at once. And that's the key part at that stage. You have a lot of cells that you can then use for your screening process. So in this figure here, the cytokine releaser cells derived from suspension storage and directly differentiated after harvesting of the cells in an unstimulated and stimulated state using 100 nanograms of LPS for 18 hours was assessed. So this is the control assay to show what happens in normal cells. Again, all this data was done in a multiplex, 7-plex from 5-plex. And then the final slide here, this shows the modulatory effect of tool compounds on cytokine secretion by macrophages upon LPS stimulation. And in this experiment, the cells were pre-treated with a high concentration of a compound for 1 hour and then stimulated for 18 hours with 100 nanograms of LPS. And after 18 hours, a supernatant was collected and the levels of IL-6, TNF-alpha, IL-8 and IL-10 were determined by the FirePlex assay. So this allowed them to go through a lot of samples very quickly by using their imager in-house to profile the cells against a range of cytokines, all in a multiplex format. So the conclusion of the work that they came up with was that FirePlex was obviously used to study the profile of cytokines that were secreted, and it's been very successful at Roche in terms of the way they've increased their throughputs. But they've shown that they managed to modify their existing process to have iPSC-derived macrophages that they can really now get good close resemblance to the primary macrophages that they had in the past. and they do recapitulate those key functional characteristics. So they can now really bulk up those cells to increase their screening throughputs. So this approach can, of course, be of use to a range of researchers who want to make detailed characterization and multi-phenotypic profiles of cell types. And so with that, I will pass back over to Will to summarize.
Thank you, Paul. So just to end this tag team presentation. So I hope you've enjoyed, and I hope you can see the processes that we go through in order to be able to generate antibodies, and then to utilize those in a couple of examples. But there are more, but there are a couple of examples, such as the multiplexing or the FirePlex. And I think it's important to understand that that's available for you, and that we can get into conversations around licensing, etc., which we have done in the past with a variety of different folk. I mentioned nanostring earlier, but there's an opportunity there to have some high quality reagent content from Abcam partnered with yourselves, either as a biotech or within the pharma environment, to be able to utilize those cell lines in your own assays. So please come and talk to us, and we would love to be able to help you. And I hope that's been a useful overview of our processes and how we can work together. All right, thank you very much for your time, and we'll move on to a question and answer session.
Thank you, Will. I think that was a very good overview of the whole process you're going through to deliver this consistency in data and support for science. I can see one question from the audience. So what if, and you tell me who it should be addressed to, what if you don't have a knockout or a knockout that is lethal?
So I'll take that one. So I guess there are occasions when the knockout either is just not available, or it's a lethal in one way or another. So I mean, in the past, what we've done is, for example, doing siRNA knockdown does work, particularly in cell lines. It's relatively easy to do, providing you're doing that validation. So in a full knockout, we would do a genomic and proteomic validation and make sure that knockout is functional. In an siRNA, we would probably do the same thing. We would align it with a Western blot, make sure that we're actually seeing a knockdown of the protein of interest in those cell lines. But the other thing to bear in mind is that, we were generating antibodies, and they were good antibodies with, some good old-fashioned detective work, understanding the protein, understanding the target of interest, knowing where that was going to be in a positive tissue or a positive cell line, knowing it was going to be negative in a negative cell line, and then comparing those two. And that relies on the literature, it relies on the evidence that we've got to us, but that can, that's a very reasonable and functional way of doing it if you don't have a knockout that's available to you.
Thank you. I'm sure that answers to the question. Other question, are the cell lines available to buy, or is it just the lysates?
So both of them are available. So there are more lysates than there are cell lines, and we started that project earlier, so you can get lysates through. But yes, you can buy the cell lines. Those will be provided to you, couriered over to you, and then you can grow those in your own lab, and we'll provide all the backup validation data that's associated with those, all the proteomic testing data and the genomic testing data that gives us confidence the cell lines are truly knocked out. Thank you.
Again, a very comprehensive offer. I can see another question. You focus on the recombinant rabbit monoclonal. Why not use phage display to generate the recombinants?
It sounds like it's all for me right now. So
you can delegate. You don't have to.
Well, it's all in my area of talk right now. So you can, I guess, is the answer to that. I mean, we have a great recombinant RAB-MAB pipeline, which generates some fantastic antibodies. We have also got phage display antibodies also on the catalogue. It works in many ways. Sometimes the affinity can be slightly different, but they're very fast in generation, and they avoid immunizing an animal. But I guess the other thing to bear in mind is it's going to also be the quality of that phase display library you start with, to make sure you get the diversity that is needed in order to be able to generate the quality of the antibody that you would get from an immunized animal. So it's a bit of a balance, but yes, they work, and there's plenty of examples that just show that they work. So you can do it.
Thank you. Another one? So, does an antibody work in all applications? Flow cytometry, ICC, IHS?
No, not in all of them. Sometimes they do, if you're lucky. Sometimes they don't. I guess it's really got to do with the antibody. Each of those applications is slightly different. Obviously, live flow has got a native representation of the target on its surface. IHC, in contrast, has been really messed up by tissue processing and formula fixation and antigen retrieval. So they're not akin to each other. So if it works in one, it doesn't necessarily work in another one. But flow cytometry and ICC tend to be quite well paired, and we often use immunocytochemistry as aligned with flow cytometry, particularly when we're doing intercellular flow cytometry to make sure we're getting it into the right cellular compartment. But you can't guarantee that one will work and the other one won't. But if they do and they're showing the same levels of specificity, then that's also a good hint that the antibody is performing well in your assays.
Thank you, Will. So I think that was a lot for you. May I conclude with maybe a more general question, so much less technical, and that's actually for me. I find it fascinating to run through this, bringing more consistency in data and reproducibility. I guess my question is, how would you say is it most driven? Is it industry driven, like pharma and maybe regulatory body who are much more demanding, and so therefore that impacted you as development partners on doing that? Or is it because it's more bottom-up approach of having access to more technologies and so wanting to just deliver better science? You can say a bit of both. I just want to point of view on
that. I would say it's probably a bit of both. We've got a large variety of customers, and we want to be able to generate the right things for all of those. And so alignment with each of those individual sets of partners allows us to be able to do that. And we've got a great network of academic collaborators, but actually when we also want to try and benefit, make sure those antibodies and those assays get into patients as assays that are beneficial to those patients. So I guess alignment with pharma partners is very helpful because it allows us to get straight to the patient wherever possible. But we've got to service all of our needs, and that includes all of our RUO academics who are available there. So it's a partnership. I guess we want to make sure we're able to be in the middle, that hub middle of being able to generate and provide very high quality reagents to people, and know that we've done all that background work to demonstrate that to those partners. That's where we want to be.
Thank you all. That makes sense. Partnership, I think it's a good word. So I can't see any more question in the chat box. So I suppose that's the conclusion of this very interesting workshop. So just a reminder, that session was recorded, so obviously it will be watchable later on the Genesis platform, but also available for OneNucleus members and broadly, so feel free to share with colleagues or other, and to get in touch with the Abcam team if you have any question. On that note,
thank you very much, and thank you very much for hosting us.
Thank you.
Thank you.
Thank you.
Bye.
Bye.